Now, in the nucleus video, I mentioned ribosomal RNA. That's just another name for ribosomes. Ribosomes are not surrounded by a membrane like other organelles. So ribosomes are in both prokaryote and eukaryote. And that's why in a previous video, when I talked about the difference between prokaryote and eukaryote, I mentioned specifically membrane-bound organelles. Eukaryotes have membrane-bound organelles, prokaryotes do not, because prokaryotes still have ribosomes because they are not bound or surrounded by a membrane like other organelles. Now, from this guy, you see the ribosome is the main structure that's going to make proteins from RNA. And here you see a microscopic image of ribosomes, and those are the little red dots. Ribosomes are made of protein and RNA. So the function of the ribosome is that they are the site or the location for protein synthesis. The assembly of the ribosomes starts in the nucleolus, which is why I wanted to do ribosomes next after the nucleus. The assembly of ribosomes starts in the nucleolus and ends in the cytoplasm. Now, ribosomes that are made in the nucleus can go to one of two locations. There's two possible places for the ribosomes to go. So number one, if they go straight to the cytoplasm, they're going to make proteins that are going to be used for this particular cell. They're going to make, let's call them domestic proteins. But some ribosomes are going to go outside of the nucleus to a different organelle called the rough endoplasmic reticulum, which I'll cover in another video. We call it also the ER, the rough ER. You can see it's basically a system of membranes, like a flat system of membranes that's covered with ribosomes. If the ribosomes go to the rough ER, they're going to make proteins, but these proteins are going to be exported out of the cell. So if the ribosomes go to the cytoplasm, they make domestic proteins. If they go to the rough ER, they're going to make proteins to be exported. So here is a diagram that shows the process of making proteins. Proteins are processed by an internal system of membranes. So we have the nucleus here, and this denser area in the middle is the nucleolus. So we have proteins made by ribosomes on the rough ER, which is this orange dotted thing. The little red dots are the ribosomes. Notice that you also see red dots in the cytoplasm. So vesicles are going to carry proteins from the rough ER to another organelle called the Golgi, which I'm going to talk about all of these things in different videos. In the Golgi apparatus, proteins are modified and they go into new vesicles. And these vesicles are going to release their proteins to the outside of the cell.